Good morning, everyone. Welcome back. Today we're going to be talking about evolution. So, evolution is defined as a change in the gene frequency of a population from one generation to the next. And that's key there, that change in gene frequency. And this is why we say evolution happens to populations, not to individuals. Because a single individual, their genes are just what they have. The frequency of those genes changes at a population level. So, for example, maybe you look at a group of insects and they all look like this top left quadrant, and then you come back and you look the next season and the insects look like this top right quadrant. Well, clearly, some gene frequency, or at least the expression of those genes, has changed. So, we say it happens at the population level. And we define a population by saying it is the same species occupying the same region or habitat. So we could say the deer population in Talladega, Alabama, they would all be deer, all the same species, and they're occupying the same region or habitat, and they can breed with each other and produce viable offspring or fertile offspring. We say frequency or gene frequency is the number of times a gene appears in the population. So, for example, we have these 16 little beetles here, these little bugs. And if, you know, 1 out of 16 expresses this black and red coat exoskeleton, but then, you know, well, we'll say in this case it's actually, 3 or, yeah, 3 with black and red. So we could say the gene frequency is 3 out of 16. But then you could come back and years later it might be 12 out of 16. The gene frequency has changed. That would be evidence of evolution because evolution is a change in the gene frequency. When we talk about the history of evolution and natural selection, two important figures are going to come up, and that's going to be Charles Darwin and Alfred Wallace. And what's interesting about them is they both discovered the same theory essentially at the same time. And they were, corresponding back and forth. And Darwin got worried because Wallace had reached out to him for his advice on some of his work. And Darwin realized that Wallace was coming to the same conclusion that he was. And long story short, due to Darwin's writings and data being much earlier than Wallace's, Wallace eventually acquiesced and let him get the credit for the theory of evolution by means of natural selection. So when we talk about natural selection, what we mean by that is that individuals who are best suited to their environment are going to be more likely to live and reproduce. So they pass their genes on to the next generation. So we call that survival of the fittest. So natural selection is nature choosing those individuals that are best suited to their environment. And here's a little image of all the places that Darwin went. Went all around the world, and in the Galapagos Islands is where, his biggest, I would say, most famous evidence comes from, and that's with his finches. And essentially what he noticed with the finches, as you see here in the top left, we have this large beaked finch, and then you have a medium beaked finch, and some smaller beaked finches, and then some with long beaks. And essentially what he noticed was that depending on the island, depending on the environment and the seeds that were available, certain finches became dominant. And maybe in that particular area, only finches with large beaks lived. And usually those were in the areas where the seeds were large seeds. And in islands with less growth, you might see finches with smaller beaks. And those finches would do well because the seeds were small. So we said the environment is nature is choosing who lives and who dies, and this is changing the structure and the adaptations of the animals that live in these environments to the point that eventually you get birds that, because their beaks are so different in shape, they have different bird calls and mating calls, and then that causes these finches to only breed with individuals of similar mating calls. And technically and arbitrarily, we would say those are two different species if they no longer recognize each other for breeding, even though it's genetically possible for them to breed. Because they don't, we could say that they are different species. And he had his famous little tree branch drawing, his I think drawing, which essentially was to show the relatedness of these species that he was observing in all these islands that he went to. And essentially the theory was that a mainland finch was the ancestor to all of these Galapagos Island finches. So natural selection example you could think of is if you have a predator, like an eagle flying over all these mice, and perhaps the surface is dark. Some of the mice are dark, some of the mice are lighter color. Well, the predator is going to notice the individuals, the prey, that do not blend in with their environment. So the lighter colored mice are easy pickings for the eagles. So they get eaten more often. They're less likely to survive to reproductive age, and they start to die off. So over time, the change in gene frequency, or the evolution, leads to a population that's mostly dark mice. So that would be natural selection. We can think of the same scenario with beetles. Maybe a certain beetle tastes better, a certain colored beetle, they like the way it tastes. Maybe the green ones are sweet, the orange ones are bitter. Well, then the predators are going to eat the sweet ones until they're all gone, and the population will start to look a different color. So the steps to natural selection, what's involved here is, one of the main things that needs to be the case is that organisms need to be different in order for this to promulgate evolution, because you need organisms, some that are more fit and some that are less fit. And those organisms that are more fit will be more likely to survive. And then also, more need to be born than can survive. If more are born than can or will survive, then those who are more fit will be the ones that do survive when push comes to shove. So the advantageous traits are preserved, disadvantageous traits die off. The most successful are reproductively successful, right? It doesn't matter if they survive if they don't reproduce. So they also need to be able to reproduce. Then this leads to these population level changes in gene frequency. And a classic example of this is antibiotic resistance that we're observing in bacteria. So what happens is we take these antibiotics and they'll usually kill almost all of the bacteria, but some of the bacteria, if we don't take all of our antibiotics, or even if we do, some of the population might have some type of trait that allows them to be immune and resistant to those antibiotics, and they survive. Well, when that bacteria comes back and they breed, they go through asexual reproduction, then the whole population will now be resistant to antibiotics. And that's actually the trend that we're seeing in the world today, especially in developed countries where we take a lot of antibiotics, right? We're giving more and more antibiotics to animals, and then we're also taking more and more antibiotics for illnesses that we could likely recover on our own. And what that's doing is that we're creating an environment that leads to these population level changes in bacteria that make them more resistant to antibiotics. And eventually, over time, the really scary thing is that we're going to get to the point where none of our antibiotics work to kill these bacteria, in which case we'll have to come up with new drugs, new antibiotics, or we'll just die from routine infections, common infections. So we also have this concept of artificial versus natural selection. I think I have, a good picture here. Natural selection is when nature chooses who lives and who dies. So the example I have here is the giraffes. For example, maybe there's really luscious fruits, but they're high in the trees. Well, only the giraffes with long necks will be able to get that fruit. So they'll be more likely to survive, and those giraffes that get less food will have less energy. Maybe some of them will die, and they won't reach reproductive age, perhaps. Well, then the population starts to look like all the giraffes with tall necks rather than short necks. We've done this with, now that's nature choosing who lives and who dies. Humans have done their own form of selection. We call it artificial selection with dog breeding, for example. Breeders breed dogs down into the specific size and shape and personality traits even of the dogs that they want. And we are choosing who lives and who dies. We're choosing who we want to reproduce, so it causes changes in these gene frequencies. They're all still dogs, but we can get a gigantic variety of dogs. And artificial selection and dog breeding, if you read Darwin's work, was actually one of the key topics that he liked to use to try to explain how organisms can change over time. And we've done this with corn, we've done this with all types of crops and all types of animals. Now, there's certain forms of natural selection and ways that a population can change based on how nature is choosing who lives and who dies. So it can affect their genotypes, and also the outworking of that is their phenotypes. Their phenotypes are actually what's selected for, because that's their physical characteristics that then changes how they actually interact with the environment. But obviously, their genotypes are determining those phenotypes. So the first type of selection is called stabilizing selection. So you can imagine a population of mice where you have really light mice, medium, fur coat mice, and then dark mice. And for whatever reason, the mice that are a medium tone are more likely to survive. Well, we call this stabilizing selection because you get to a medium or a middle phenotype. That becomes more pronounced. Directional selection is like what we saw with the giraffes, right? The population moves in one direction, from short necks to long necks, from small squirrels to large squirrels, from small wings to large wings, or large wings to small wings, just in one direction. We call that directional selection. And then diversifying or disruptive selection is when you have two different, very different shades that are selected for equally. So, for example, you have dark and light moths. Well, those that are really, really dark do well and survive, and those that are really, really light can do well and survive. So nature chooses the extremes, and then those medium shades end up dying off. And then here's another example of that with mice. So evolutionary change can occur in a few different ways. We have mutations. We have organisms just choosing a mate leads to evolution, because certain organisms that are chosen to mate more often, their genes are passed down more often, and it makes that change in that direction of that gene. So we see a change in gene frequency. So mating is actually rarely random. It's sexually reproducing organisms. So we have this mate selection that's going to have a huge effect. But just we'll talk about mutations for a second. A mutation just means a change in the nucleotides or the DNA sequence, and that can lead to phenotypic outworkings or changes. So, for example, you might have genetic mutations on a fruit fly that could change its eyes from, red to white, and that might be from a mutation rather than from some type of gene shuffling. We have two types of sexual selection. We have intrasexual and intersexual. Intrasexual selection is competitive interactions between members of the same sex. So, for example, two bucks, males ramming horns and trying to compete for a mate. So the males are competing with the males to attract a female. Intersexual selection, which we say is mate choice or female choice. So this would be competition or interactions between the sexes rather within one sex. So, for example, a peacock showing its feathers and trying to attract a mate, larger and larger horns of certain cattle to try to attract a mate, things of that sort. And the sexual selection leads to what we call sexual dimorphism, or two types of sexes, two morphs or appearances. Because the longer that sexual selection goes on, the more pronounced these differences can become until they look very, very different. So, for example, here you have these males that have these bright colors and females that have a more muted color. Because there's really no reason for the female to have these intricate colors, because it's the females who are choosing the males. So the males are just trying to attract the females with their bright colors. We could see it with males with horns, or you'll see something on a male animal that just looks weird and useless. And it might have a function, but it also might be extremed, or really extreme, because it allows them to attract a mate better. And we see that with horned animals and all sorts of things. So some other things that can change or lead to evolution, one of them would be migration. So we think about immigration and emigration. When emigration is when individuals move out of an area. Immigration is when individuals move in. So, of course, that's going to change the genes that are in that population and the gene frequency, right? If you have 10 orange ladybugs and then you have 30 black and red ladybugs move in, well, now the gene frequency has just been completely flipped and changed. So, of course, that could lead to evolution, which just means a change in gene frequency. We can also call this movement of alleles. We call it gene flow, that the genes are flowing from one place to the next. A change in allele frequencies that occurs by chance or accident we call genetic drift. And there's two examples of this. We have the founder effect and the bottleneck effect. For the founder effect, this would be a type of genetic drift in which a small number of individuals found a new and separate population, or they like form a new and separate population. So for example, we have all these bunnies here. Some are gray, some are brown. But then only the gray bunnies move to a new location. Well, now this gray bunny is the founder of a new population, and that new population is all going to look gray. So we have this change in the number of alleles and the frequency of these alleles because we have this founder effect, this new organism that, or group of organisms in that species that have founded a new population. Their genes are different than the genes that they came from. And they saw this with the Afrikaners, the Dutch settlers in South Africa. A big group of them moved down to South Africa and tried to colonize it. And it was a pretty small group, so they didn't have a large genetic diversity. And what actually has happened there, because of this founder effect, is that their reduced genetic diversity has led to a higher frequency of certain diseases because they don't have the genetic variants that protects people from duplicate bad genes on chromosomes. So even schizophrenia is at a higher rate in this area among these Afrikaners and descendants of these Dutch settlers. The bottleneck effect is another example of this, and this is when I literally just, for those of you who are listening, we have this bottle with all these different color beads, and the bottle has a neck that's more narrow than the base of the bottle. Well, if you pour just a few beads out of that bottle, you might get one of every color, or you might just get red beads. So you're losing all these genes because you bottlenecked. There's a bottleneck event that reduced the population to such a small number that you also reduce the genetic variability. And a great example of this is the northern elephant seals, which in the 19th century, they were hunted because their blubber was really good for lamp oil. It's oil rich. So people would just hunt them like crazy, and their population got all the way down. By some estimates, they think it got as low as 10 to 20 seals left. Then they started to be protected. There was more awareness, and now their population numbers have come back up, but their genetics were greatly reduced. And that brings us to the topic of microevolution versus macroevolution. And a good example of this, like micro, we think of microscope. This is really, really small evolution. Macro is big. We think big changes evolution. So microevolution is usually going to occur short term in something that you could just observe, right? You could just take some dogs, breed them down until you got dogs that looked a little bit different. That would be microevolution, small changes in the short term. Macroevolution is large changes. We're talking about novel designs over thousands and thousands of years to get large changes that build up. And really the idea is that macroevolution is a lot of microevolution just over a long period of time, so that when you look at it, you see much larger changes. So macroevolution is when we're talking about speciation. Microevolution is when we're talking about changes within a species, usually. So an example we have for microevolution is beetles changing in their colors over a short period of time. Maybe they're green and brown, and then they move towards more green. Still beetles, small changes. But for dinosaurs, for example, all the different dinosaurs we see, in order, if they did share a common ancestor, then it would take a much longer period of time to get that diversity of animals and all these different species that we observe. So when we're discussing these big changes in new species, we're talking macro. When we're talking small changes, usually within a species, we're talking micro. We'll talk about a little bit of evidence for each. So one of the pieces of evidence for microevolution are these tiny little guppies. And with guppies, they have pools that are below waterfalls and pools that are above waterfalls where they live. And for example, if we say in a pool below the waterfall, it's a smaller area. These animals are smaller. They reproduce early and they don't have bright colors. And in the pools above the waterfall, they're larger, they reproduce later, and they're brightly colored. And you would wonder, well, why are these guppies, don't have any bright colors at the base of the waterfall, but above the waterfall, they all have bright colors? Well, in this case, below the waterfall, there is a predator, and above the waterfall, there is not a predator. So where there is not a predator, the guppies have these beautiful colors, and they have these colors to attract a mate. But below the waterfall, they don't have these beautiful colors because the predators will notice them easier and eat them. So these guppies have had these small changes. They're still guppies. They can still breed with each other. They've had these small changes, though, these changes in gene frequencies, due to the presence of a predator. So we would say that is microevolution. That's not huge changes. It's small changes within these species. And then you could say, well, if you introduce these species back together of guppies, and now they don't even recognize each other, genetically maybe they could breed, but just by appearance and their behavior has changed so much that now they don't want to breed with each other, well, then we would say they've evolved into separate species. So evidence for macroevolution, we'll talk about the fossil record, comparative anatomy, homologous structures, vestigial structures, and gene sequencing. So as far as the fossil record goes, you do see a trend in simple organisms to more complex. So if you're trying to form a large-scale theory for how these organisms came to be, then the fossil record, you could look at, similar structures from these ancient animals to try to see how they might be related to each other. Homologous structures, a homologue, this would be a similar structure in different organisms. So this could be evidence of a common ancestor. So for example, a human and dog, maybe they do have a humerus bone, and it's built similarly, and it's in different organisms. So these different organisms having the same structures could be evidence that they once shared a common ancestor. And it's a little bit harder to imagine with organisms that are so different. But if you take organisms that are more similar, like humans and chimpanzees, then these homologues, they even look more similar. But even with animals that are very different, they'll still have these similar structures in these very different animals. Now, analogous structures means it's the same function but different structure. So, for example, bird wings, insect wings, and bat wings, they are all for flight, but they're not the same types of structures. They're analogous in function, right? But they are not homologues. They're not built the same way, even though they're all doing the same thing. So none of these are evidence of common ancestry because they're not built the same. They're not the same structures. They're just analogous to each other. And then vestigial structures, which some of these are, they'll say they're vestigial, and then there'll be some evidence that there's some functions for them. But a vestigial structure just means something that has been reduced in size and function over time for so long because the organism was no longer needing it as it adapted to its environment that it becomes unnecessary for the organism. So this could show relatedness between different organisms. For example, certain bones and whale flippers. Manatees have fingernails on their flippers. And then for humans, we have what's called this third eyelid, which is this tiny little organ that has been reduced, which used to be a third eyelid. And it's crescent-shaped, fold in the inner corner of the human eye, and it does help drain tears and remove debris. And then, of course, DNA, RNA, and protein similarities could be evidence as well. If you look at organisms that look similar, then they have more similar DNA to each other, usually. So, like humans and chimpanzees have very similar DNA, but humans and tomato plants, it's not very similar. So, the more similar the structure, the more similar the DNA, and the DNA is the blueprint for the structures. So, it makes sense, right? If you looked at two skyscrapers that looked similar, their blueprints would also be similar. So, it could be evidence of relatedness. And that's it for this lecture. Thank you.